Hello everyone. Today I will discuss about history of pharmacognosy and modern perspective. So whenever we talk about history of pharmacognosy, we can easily say that history of pharmacognosy as old as human civilization, because as you considered from the very first day of human civilization or mankind, people were totally dependent on nature for their food habit, medication, and other purposes, where different sources, more specifically forest resources, which actually provided enormous plant products to the entire mankind from that particular era, it is of greater value. From the first day of human civilization, we started our journey along with the plant products. We started believing on those products as medications too. As time passes, our thought, our belief, our trust towards all those products became stronger, even more stronger day by day. Country like China, India, Egypt, Greece was involved in the medicinal uses of plants even long before the beginning of Christian era. Before Christian era, people started their journey along with the plant products and other medications like acacia, castor oil, and fennel. Apart from that, people also introduced along with the product like iron oxide, sodium chloride, sodium carbonate, and sulfur. Most of the medicinally active substances identified in the 19th and the 20th century were used in the form of crude extract. The oldest known herbal is Pensao, was written by Chinese Emperor Shen Nung around 3000 before Christ. It contains 365 number of drugs, one for each day of the year. It means it was a process, it was a discipline, so that people started taking the natural product of natural origin every day in limited doses, in limited amount. In India, if we consider Charaka and Susruta, they played an important role too. They introduced Charaka Samhita and Susruta Samhita. Charaka Samhita was introduced by Charaka and Susruta Samhita was introduced by Susruta. If we consider today's modern medicine system or allopathic medicine system, the concept of medications and surgery that comes from Charaka Samhita and Susruta Samhita respectively. Charaka Samhita is it talks about actually the medications, the medicines of different aspects, which is used for curement of number of diseases, whereas Sushruta Samhita mainly talking about or focusing on the surgery. It means the modern medicines rooted behind in the form of Charaka Samhita and Sushruta Samhita. Charaka made 50 groups of 10 herbs, each of which, according to him, would suffice an ordinary physician's need. Similarly, Sushruta arranged 760 herbs in 7 distinct sets based on some of their common properties. A large portion of Indian population even today depends on Indian system of medicine, Ayurveda, or an ancient science of life, where they also believe that Charaka Samhita and Sushruta Samhita are the pillars for today's medical system. A number of renowned physicians, chemists, philosophers, and scientists had an immense contribution in the field of pharmacognosy. They are as follows: Hippocrates, very well known name to all of us, who is considered as the father of medicines, 460 to 360 before Christ. Aristotle, 384 to 322 before Christ. Dioscorides, 40 to 80 before Christ. Galen, 131 to 200 before Christ. The early Arabian physicians, there was a period of approximately 1000 years during which little, if any, progress was made in the medical sciences. The first pharmacist, Galen, was known to have had a number of pain relieving materials, including opium, in his apothecary. For the next few hundred years, the formulations of the medicaments in use changed very little, although some bold attempts were made by chemical entrepreneurs, Paracelsus, who developed mineral salts, which might have had the potential of being universal curative agents. However, throughout this period, the use of herbal extract for medicinal purposes never lost its place. The importance of the extraction method and alcohol as an extractant was reported by Lemery in between 1645 to 1715. William Withering in 1785 published an account of some of the medicinal properties of foxglove leaves based on 10 years of experimentation. Percolation process was used for the crude drugs. Columba and alkaloidin drug became official in 1788. In 1803, the French pharmacist Diroson isolated nicotine from opium. In 1806, Turner isolated morphine from opium, and its role in alleviating pain was recognized. In the next few years, strychnine, emmeline, brucine, piperine, quinine, colchicine were isolated. The French pharmacist Pelletier first reported the isolation of strychnine and other products from ignious beans and later from Nux vomica seeds. A few extraction process for alkaloid was developed by Stas and Otto in 1852. Other important discoveries during this period were the isolation of nicotine from tobacco leaves, cocaine, pilocarpine, ephedrine, podophylltoxin, etc. Isolation of ergotamine, digoxin, reserpine, theophylline, and quinidine are the significant discoveries of the 20th century. Pharmacognosy, in its modern context, is a dynamic field that integrates traditional knowledge of medicinal plants with advanced scientific techniques to discover, develop, and utilize natural products for healthcare. It encompasses the study of the physical, chemical, biochemical, and biological properties of natural substances, primarily from plants, but also from other natural resources like microorganisms and animals, with a focus on their medicinal potential. Modern pharmacognosy leverages tools like phytochemistry, microbial chemistry, and advanced analytical techniques like LCMS, GCMS, NMR, etc. to identify, quantify bioactive compounds and explores their pharmacological activities. Apart from this, several advanced techniques, more specifically chromatographical separation methods like thin layer chromatography, HPLC, HPTLC, gas chromatography, paper chromatography, column chromatography are of greater importance. Besides this, the usage of UV spectrophotometer, infrared spectroscopy are of greater value. The involvement of camera lucida and electronic camera microscope with high efficiency plays an immense role to identify the microbial characters lying within the plant products. Whatever product we are choosing, we have to go with the dissection of the given sample to identify all those characters properly. We need to have higher efficiency camera microscope. To dry a proper product, the proper usage of lyophilizer is needed also. Therefore, in today's modern perspective, pharmacognosy not lying along with the traditional concept It maintains its own features along with the modern technologies, modern aspects, modern subjects, and many more, so that students and others can explore, or researchers or any of the applicants, they can carry their research work, study projects by the help of those associated tools. Thank you very much.